and we have a long day. So it's with a great pleasure for me to introduce to you our next speaker, who's going to talk about Therapeutics 101. So he's going to dive a bit deeper into all the therapeutic opportunities for the different pillars. And this is our very own head of research and discovery, Dr. Ed Weeber, who really was one of the biggest impetuses to starting the research program at FAST. Back in the early days when FAST was discovered. And he has been part of FAST well before my time. So I am only a product of Dr. Weeber. He has taught me everything I know about neuroscience, which you can, if you criticize what I know, it's his fault. If you are proud of what I know, it is also his fault. But this is Dr. Ed Weeber.
Awesome. Hello. I think you all have your coffees. You're warming up your hands now. Welcome to sunny Florida. Florida's the only place I've ever lived where I'm colder in the summer than in the winter because it keeps everything so AC. But I'm really glad to be here and be able to talk to you. So I really struggled with this talk. Introduction 101, I started making the talk and thinking how I could compress 10 years of education of neuroscience and cell biology and genetics into a 20 or 25 minute talk. And I thought, well, maybe I should step back a little bit. So we're going to take a little bit of a different view, kind of a 30,000 foot view, and talk about understanding the pipeline, understanding how the pipeline for therapeutics occurs. And I think it'll put things more in context when you start looking at the talks. And I think that Allison did a great job of an overview, perfect on the neuroscience, Allison, and talking about the different pillars. And I think if you can kind of keep that in mind when you're going through the talks today, you can certainly be able to see in that context where all these different scientists are doing their work, where they're exploring, and where in that therapeutic pipeline it is. So when we think about the long road from bench side to bedside, this is what we're talking about, a laboratory bench, right? And so everybody has this idea in their mind that science is very clean, and you have these researchers that are pouring different colored liquids into different tubes. It's actually quite messy, and so I'm going to peel away some of that mess and complication a little bit and talk about how therapeutic pipelines are kind of like that bench. Right? It can be very messy, it can be very complicated, but it follows a certain set of rules. But it's going to be different for each one of those pillars, and each one of the scientists that come up here and talk about it are faced with different challenges as well. And if you look close at that picture, you can see there's little green tape on that bench. For the scientists in the room, a lot of times that tape will delineate where your piece of the bench is. So if you have a lot of people working in a laboratory, that may be your space, right, where you do your work when you come in between classes and whatnot. So like I said, it's very messy, but this is how we do our work. So when you think about basic science research, and I think Allison talked a lot about the biology of AS, the genetics behind AS. When you think about that basic science research, it involves a lot of different people, right? So I've got listed here geneticists, molecular biologists, neuroscientists, people who are experts in animal behavior, biochemists, cell biologists. We're all scientists, but we all look at things a little bit differently. We all speak a little bit of a different language, and it's great to have all those different perspectives when you're doing that basic science research, and especially in Angelman syndrome. So what is the therapeutic pathway or therapeutic pipeline that we talk about? It is very systematic, but again, it can be a little bit messy, and it can change in a lot of different ways for what academics can do, which is really where the research starts, and then gets really handed over into industry that has the expertise and the breadth of expertise to carry that through and do all the proper work to get it into patients. So when we first think about identifying therapeutic targets, and you're going to hear a lot of talks about those targets and how they fit into those different pillars, it really starts off in the basic science research looking at that, identifying biological targets, and then screening for those therapeutic targets. So when we start talking about target identification, identifying where those biological places are that we can insert therapeutics to treat AS, it really comes down to this little mouse right here, this UBE3A mouse. And I've actually got and Art showed this too, this is the PubMed citation that came out in 1998 for this particular mouse, right? And to go back a little bit in time, back in the 90s when they were first starting to create mice and mice models, there weren't that many of them out there, and there were even fewer out there for neurological disorders. And so this was really a unique mouse model, and Art freely gave this out. Now, the models that were out there, there were some of them that were out there for Alzheimer's disease, and that went through decades of litigation on who owned that mouse, who was able to use that mouse, who was going to profit off that mouse. So I started at Vanderbilt in 1999. When I left to go to, I started at Baylor in 1999. When I left to go to Vanderbilt, I went to Art and I asked him, can I take some of these mice and start my own colony when I go to this other university? And his response, I went out on a limb to ask him this. His response was, anybody who wants to use this mouse, it's freely available to him. He went beyond what the monetary value was of this mouse because he wanted to drive the research. And his free use of all the tools and the knowledge that he created way back then is really what got all the geneticists and biologists and neuroscientists to be involved in AS, to come up with those biological targets that we're going to talk about today. I don't see Art, but I do want to acknowledge his contribution because we stand on the shoulders of great people, and he is one of those great people. And I want to give him another round of applause. Really. He was amazing. Standing ovation. This Art is, it's incredible to have him here. He's not in the room, but I think it's important to acknowledge, he talks about all the contributions of all these other scientists, and we really stand on his shoulders. And so the first use of this mouse model that he created, and we still use it today. There's still publications coming out that are a direct lineage of the mouse model that came out over 25 years ago. But we were able to identify some of those biological targets, understanding the silencing of the gene that he talked about, understanding what some of those targets are, what UBE3A actually does, what it ubiquinates, the targets of potential intervention, like what is going on with the the ATS, the anti-script. The molecular and biochemical changes were hugely important. Those were really what the first targets of AS were. And the first gene therapy that I did with an AAV was in this particular mouse model, and Art's back now here. So you can thank him if you see him later on today. If you go to PubMed, and now we have this beautiful literature repository on the website, I really suggest you go to it. It's very interactive and it's very, very cool. But if you look at Angelman syndrome and therapeutic, if you put those two terms in, you can kind of see how the increase in those publications have occurred, over the last 20 years. It's really amazing. And this isn't just the same scientists publishing more papers. These are more and more different types of perspectives coming in and publishing and identifying those targets, some of those that are going to be talked about today. I think that Allison did a really great job talking about those tools in the biorepository page that's on the website as well. There's a lot of tools out there. We've been developing tools for a long time, and FAST has been very, very integral in making sure that those tools are available to everybody. And I can tell you right now, there are models that are being made that aren't being shared. And so FAST has been the one there to make the tools, to make them available to everybody and all the researchers to go through this pipeline and do this target identification and validation. So we already know what the targets are. We start to evaluate those targets. We can do this through a lot of different ways: genetic studies, biochemical studies, using cell models or animal models. And really start thinking about the targets in each of these pillars. And so when you think about a gene replacement or enzyme replacement or using HSC, these are really platforms, right? This is where the research is going in understanding these platforms and how to figure out we can use the tools in the animals to do what we call a proof of concept. but they have to get moved into a patient somehow. And so there's challenges involved in that. But if you really think about targets, and targets that have been published on, really pillar 2, that turn on that dad's gene, where on the ATS do you target? Do you target the RNA or the DNA? Do you just target how much you can turn up the dad's gene that's there but silenced? So there's a lot of different targets that go into pillar 2 as well. And then pillar 3 is more along the lines of a traditional type of therapeutic discovery, right? So any drug that you take, any prescription that you get has to go through a really defined way to go through the pipeline to make sure that it's safe, it's hitting the target that it's supposed to, and then it goes through all those processes to get to clinical trial. So now when you start moving from identifying biological targets to going into screening those potential therapeutic agents and then kind of refining them, that screening takes a lot of work. And again, it's different for each of the pillars. And if you think about pillar 1, we throw out AAV. This is an adeno-associated virus. There's a lot of flavors of AAV, and they attach to cells in different ways. And they can deliver things in a lot of different ways. So when we think about AAV and all the things that you need to do to make that AAV not only safe but effective, you have to think about which isoform do you use, how do you package it, what promoter do you use, which AAV are you going to use. The same thing with enzyme replacement therapy, and Barbara Bayless is going to come up here right after I talk and speak about enzyme replacement therapy and the challenges that are associated with that as well. With pillar 2, sites of ATS interference. All of these have to be validated. These targets have to be validated. You can do it in the mouse, and the same thing with pillar 3. You can do these in the mouse. But here's the challenge with the mouse that you probably haven't thought about, is that as great of a utility as that mouse model is, all those mice are genetically identical, okay? So if you come up with a target and you're trying to validate that target, it may work really great in a particular mouse model, but if that mouse model is all genetically identical, it's going to look the same in every single mouse you treat, right? And this, when we talk about hit-to-lead optimization, this is where it really becomes a challenge. So in a different strain of mice, in a different, any kind of genetic variation, that target may not work as well, or that therapeutic may not work as well on that target. So how do we get around this? We get around this by doing human iPSCs, and there's going to be a couple talks today about the iPSCs and the initiative to make a lot of these iPSCs. And what is an iPSC? They're induced pluripotent stem cells. So they're somatic cells that we can get from a patient. We treat them so that they look kind of like stem cells, which means that they're immortal. We can grow up a whole bunch of these cells. And then we can also treat them with certain chemicals to turn them into and differentiate them into different types of cells, including neurons. All right, so here comes the genetic variability. Now we can look at UPD, deletion, mutation. We can look at all the different flavors of AS and validate those targets and see how well they're going to work on all those different types and genotypes of AS. It's really a great system. And again, FAST has been involved in making these iPSCs, making them for all the researchers that are out there, and they're in the process of getting banked right now. And then also preclinical studies, right? So this is where it starts to transition into the talks you're going to hear about and going into clinical trial. And this is really where the industry partners have to come into play. So anytime you're targeting a therapeutic, it's challenging, but when you're targeting the brain, it's especially challenging because you have to get past this thing called the BBB, the blood-brain barrier, right? And it's a very, very highly developed, stable system to allow blood flow into the brain to deliver oxygen and deliver things that are supposed to get through, but it keeps out toxins, it keeps out bacteria or viruses, any kind of foreign agents. It also keeps out a lot of those therapeutics. So it's really hard to get across that blood-brain barrier. So a lot of work when we start talking about route of administration, how do you get the therapeutic in? And that's another aspect of this that you all should be kind of aware of. And then finally, those preclinical studies that after target validation and optimization, you're doing those preclinical studies. This is where really the rubber hits the road before it goes into patient. And this is really difficult to do in a laboratory. Some of this can actually be started in the laboratory, and some of the talks are going to be discussing this, but if you were at any of the talks yesterday, we threw around terms like pharmacokinetics and pharmacodynamics. These are all things that have to be figured out before something goes into clinic. And I think a lot of that will be discussed tomorrow when the industry comes in and starts talking about the clinical trials. Formulation development, how well can you make it? Is it going to be bioavailable? Is it going to work the way you think it's going to work in the laboratory when you start going to the patient? All of those things are very important. So what I wanted to do, instead of going through the specifics of the science, and listen, the whole science team is here. The room is filled with scientists. Come and find us if you have questions. Write them down, and we can go through the nuts and bolts of what an adeno-associated virus is, what an HSC is, how the antisense works. What the hell is an antisense, and why is it there? Those are the questions that we can talk through and talk about, so you have a really better understanding of where this therapeutic development is, especially in the talks that you're going to see, and where the clinical trials are and what they actually mean. You're going to have to be making these decisions for your own kids in the not too distant future. So it's a good idea, and I think Allison discussed this, it's a good idea to get a solid grasp on the science, because when we start talking about these therapeutics, you're going to want to know what it is, what it means, and how it's going to affect you and your family. So what are the targets and platforms of each pillar? We've nicely broken this down from pillar to pillar as the people are going to come up and give their talks, to kind of keep this in your mind: mom's gene, dad's gene, looking at downstream targets. Is there proof of concept for the data? Are they using the tools that have been developed? And I know Allison said no more tools. There's a lot of tools out there. So they use these tools to see, is there a proof of concept? Is it actually going to work in the tool set that we've developed before it goes into the patient? What are those tools? Are they looking at animal model, in vivo model, which is huge in the in vivo model, but it doesn't have that genetic diversity, right? So a lot of times they'll go into the iPSC cells if they can do that. And what is the route of administration? How are they getting that therapeutic to where it needs to go, which is in the brain and in the neurons of the brain? And I hope that I've given you a general idea, starting with basic science research, identifying the targets, working out the biology of AS, and then trying to find out where those therapeutic targets are and how we can exploit those to develop good therapeutics that can go into the patient. So you should know approximately where they are in the pipeline. And if there's any questions about this or any of the talks, again, we're here for you. This is why we have the symposium. And any questions that you have, we'll be certain to answer. For those of you in the back that can't read this, there's two scientists talking, and the one scientist says, the scientific purpose for keeping a head alive in a jar is keeping a head alive in a jar. And I know it's not that funny. So I'm a little bit geeky. It's not that funny, but I want you to think, thanks. What I want you to think about is that you're going to be seeing a lot of science, and you're going to be seeing a lot of data. Just understand that it's not just being done for science sake, all right? All of the data that you're going to be seeing and all of the experimentation and how they're doing it, it all works toward a goal, right? We're all trying to get those therapeutic targets validated and pull them through the pipeline as quickly as possible. And this is what FAST is here to facilitate, not just in monetary means, but also help synergize and get people together and look at their science and help them get the best science out that they can. All right, it'll be a fun day. And again, I'm here to answer questions, and everybody from the science team is here to answer questions as well. And thank you all for being here today. Thank you.